you're all invited to my place afterwards, 6 o'clock. We're hosting a potluck for Bill, and if you haven't had time to get anything together to bring, don't worry about it, just show up. We've got food and beer. There's the address, there's my cell if you need to get in touch. We'll pull out some cars from the driveway, so park there if you can, or in our yard. Don't park in the neighbor's yard, especially the one with nice grass, because he really loves it. All right, there's that. Dr. Bill Sutton is an assistant professor in the Department of Agriculture and Environmental Sciences at Tennessee State University in Nashville. Bill got a BS from Wheeling Jesuit University, his MS from Marshall University, and then he got a PhD in Environmental Sciences and Natural Resources from Alabama A&M. After that, he did a postdoc at University of Tennessee, and then one here at Clemson. So Bill was... I was fortunate enough to get him to come here shortly after I was hired. So one of the best pieces of advice I got when I got this job was use part of your startup and bring a postdoc. And so I talked Bill into coming. Best decision I ever made. He was a critical part of my lab in terms of helping write papers and grants and advising students. So I can't thank you enough for that. Now Bill is working on wildlife population and community level responses to disturbance, species distribution modeling, zoonotic diseases. He collaborates widely with a variety of institutions and agencies throughout the Southeast, and in his spare time enjoys fly fishing, hiking, trail running, and daydreams of owning a brewery someday. Now, what he did not put in that list of hobbies is that he's also very good at ping pong. We played several games last night. I think I only won one of them. So if you're there tonight, Bill says he'll take on all challengers.
Yeah, bring it.
Turn it over to you.
Thanks, Kyle. And first off, I want to thank everyone for the opportunity to be invited to come back here. As Kyle mentioned, he said he had to convince me to come here. I was ready to come, and I really enjoyed my time here. I was only here for about a year, but I really enjoyed the department, and it's really nice to come back to see how it's changed and to see who's still here and kind of see how it's grown. So thanks again for the opportunity. So as Kyle mentioned, I have very broad interest just in general wildlife ecology, but I'm going to kind of focus the talk and kind of focus around the idea of spatial ecology and spatial scale. And so I'm going to talk about it from the context of some of my research examples that go on in my laboratory. And so we'll kind of talk a little bit about some of my research interests initially, and then I'll kind of let you know what's been going on in my lab since I left here in 2014. I've been at TSU now for about 6 years, and so I've been able to start a small wildlife program there. And so I want to kind of cover the context of what I'm talking about through spatial scale. And so, as Kyle mentioned, some of my basic research ideas, I work kind of in the context of global change ecology, and so I kind of work in sort of three primary areas. I work in climate and habitat change, and also looking at the impacts of pathogens on wildlife. These are all, of course, of the context of what the impacts of these disturbances in terms of climate change and land use, and also pathogens interact synergistically to have impacts on wildlife populations. Now, as you'll see today in my talk, spatial scale is kind of an important thing in my research. I work at a variety of different spatial scales to answer a variety of different questions. But, however, the larger realm of my research kind of sits in this sort of envelope of just basic conservation or applied conservation biology. And so I work with a variety of different wildlife, but my examples today are going to be primarily herpetofaunal, because that's kind of where I got my start. You'll have several salamander examples with a little bit of some extra stuff at the end. I'll kind of get into some of the project that I actually worked with Kyle as a postdoc here. We've almost finished it. We're getting really close to it now, which is great, which is kind of sad now because it's a long time, but that's the way it goes sometimes. Okay, so one of the kind of seminal papers that I've had to think about as a graduate student and kind of starting my lab is this paper by John Wiens. It's called Spatial Scaling and Ecology. It was published in 1989, but it's extremely important from understanding the idea and the consequences of spatial scale when it comes time to determine what spatial scale you're going to do your research and what types of questions you can ask with that. Because as you may have learned in some of your courses, that there's two terms called grain and extent, and grain represents kind of the base, or the kind of as far down as you can go down with your data, whereas the extent represents the scope or kind of the top end of your study area. And so there's a really clear relationship between grain and extent in terms of what you can answer with the study question at hand. And so as we get down on the lower end of the scale here, we can tend to focus on more mechanistic or kind of understanding mechanisms of small scale study areas, as opposed to as we get to the larger spatial scale, we start to generalize more on things like larger disturbance patterns, disturbance regimes of ecosystems, and larger patterns of things like ecoregions. So kind of understanding this relationship between grain and extent is extremely important, not only for developing a study, but also understanding how far your data is going to take you and what kind of inference you have based upon that data. And so, for example, we may have a study that we're working on right here that occurs somewhere between this grain and extent. That we may know some kind of mid-level type of an estimate where we may be able to kind of derive some larger ecoregion scale things, but maybe not quite as large as the larger landscape. And so what I'm going to talk about today is kind of how my research sort of fits into these different spatial scales and give you some examples of kind of what we've been working on at TSU since 2014. And so throughout the presentation, I'm going to kind of keep you on track. I sort of had this sort of diagram that's kind of adapted from Johnson's 1980 publication talking about the different kind of animal spatial scales and also between John Wien's manuscript to kind of give you an indication of what spatial scale this is really going to occur at. So I'm going to talk a little bit about some of my field experiments that have kind of operated more at the microhabitat scale, at the home range scale of the organism in question, or at the larger landscape across multiple populations. And so we'll kind of use that as our guide to stay on track. All my examples are going to be kind of through the lens of my laboratory. I pretty much use guided research with graduate students, so that's a lot of the way in which I do it. So even though this is research that's occurring in my lab, there's people that are actually doing this stuff. Believe me, I'm not doing all this stuff. I just try to guide the students and help out as much as I can. I've had some great students that have helped me out along the way. And so the first study I'm going to talk about, was by Nicole Witzel. Nicole will be graduating this summer. And Nicole's project, when she first started off, she was telling me she had an interest in both. She had an interest in basic molecular ecology and also with salamanders, and had an interest in environmental DNA. And so it just so happened that while I was talking to a professor at Middle Tennessee State University about this animal that occurs near the Nashville Basin, it breeds in aquatic environments, and we thought about trying to develop an eDNA protocol to detect this animal. Well, this larger group of salamanders you're looking at right here, this is the family Embystomatidae. This refers to the larger group of the mole salamanders. These tend to be animals that burrow underground. They have a really unique lifestyle where they breed in these temporary ponds that are fishless. And so they'll target these areas, move in mass migrations in the fall or spring, and lay eggs all at once, and then leave the ponds. Well, the idea is that the larvae can hatch, grow without pressure from predators, and be able to metamorphose before the ponds dry out. And so they kind of specialize in these kind of temporary environments. Well, these guys right here are all pond breeders. And you might recognize a few on the screen here. This is the spotted salamander, mole salamander, marbled salamander, and tiger salamander. All these species occur in Tennessee. But there's also two species that are really similar. There's one on the left here is called the smallmouth salamander, Embystoma texanum, and Embystoma barbari, which is the streamside salamander. Now, based on external appearance, they look extremely similar, and genetically, they're also very similar. However, the breeding habitats are really different. This species on the left breeds in these temporary pond environments, whereas Embystoma barbari, being called the streamside salamander, breeds in very temporary, ephemeral stream habitats. And so they're also going for the same kind of strategy where they're targeting these habitats that don't have predators. However, the problem with this is that these streams only hold water through about 6 months of the year, and during the summer season, when a lot of streams are actually evaluated for whether or not they're a stream or not by local conservation groups and development groups, they aren't targeted. They're actually not classified as streams. They're classified as what is known as wet weather conveyances. And so that makes that stream really open to development and being developed into housing developments and things like that. And so they actually have a presence. This species is actually state endangered in Tennessee. So we wanted to develop a rapid protocol to where we could go out and actually go to a site, detect if the animal's present, and then potentially get protection in place if we could do that. And so Nicole took on the monumental task of trying to develop this eDNA protocol for the streamside salamander. And I say this because I was talking to some of the graduate students earlier, and this is one of the projects where I almost kind of had a big error because I had done an initial eDNA project with hellbenders. It seemed pretty straightforward, so I thought, yeah, I can do this. I can develop an eDNA protocol. Well, it turned out it's much more complicated than I had really planned. So the two questions we wanted to evaluate was, can we develop an eDNA protocol that's specific to streamside salamanders, and can we take this and actually maybe potentially predict some estimate of biomass at a site based on this protocol? And so to give you an idea where the streamside salamander occurs, this is Davidson County. This is where Nashville is. Murfreesboro is right down here. So you can see the range kind of skirts north and south. This is disjunct from the larger distribution of the species, which occurs up here in Kentucky and also in West Virginia. And so we're kind of dealing with this sort of rapidly developing area of Middle Tennessee. And so folks who aren't maybe familiar with environmental DNA, it's basically DNA that's shed by an organism into the environment. It can be in the form of multiple things like tissue, fecal material, sperm, egg cells, just basically anything that an organism sheds. Really what it comes down to is, are you able to actually harvest that data, or harvest the DNA, be able to use it to actually develop a protocol to detect the species? And so, of course, it's really useful for detecting presence-absence, but as you kind of try to use this to go further with it, it presents some problems with trying to actually tell more about just presence-absence. And so for this study, we're trying to target the streamside salamander DNA when it's in water. So that's kind of the point of the project. Now, this slide I'm showing you right here, this slide basically took Nicole about 1.5 years to get. And so this is basically the protocol finally showing that we got really good amplification from the eDNA protocol. We actually did several dilutions to show that as we reduce the dilutions over time, we get kind of more less concentrated dilutions. We're getting a really nice kind of estimate on our quantitative PCR. The x-axis refers to the number of cycles, and so if you have an amplification that occurs further up along this, that means you have more DNA in your sample. So it gives you kind of more of a quantitative way where you can estimate copy numbers of DNA as opposed to just traditional presence-absence. So you can use it for both. And so after about 1.5 years and trying about, I think, between 10 to 12 different protocols, we finally found one that worked. It was very specific to the animal we wanted, and it also excluded all the other congeners. Now, one thing you could say is that the other species don't breed in streams, so why should we care? Well, it's also really good to show that there's no chance that you might have DNA from another species that's falling down into the stream where you're detecting the species in question. So we wanted to make sure it was both specific and sensitive. And so when Nicole started this project, we kind of developed some general hypotheses, given the fact that if this was in fact going to be used for estimating some estimate of biomass. And so what she predicted is that in early December, the adults moved to the streams, and from about December to January, the males and females will breed, and they'll lay eggs almost immediately, and the eggs are fertilized and ready to go. Egg hatching begins somewhere right around between January and February, so right around now we're getting larvae that are hatching. And then as you see, the amount of eggs decrease over time, and then we see the larvae start to metamorphose from the stream right around May. Now, Nicole kind of made a prediction that she thought she'd see a kind of a nice peak in DNA somewhere between January and February, in which that would decrease over time. So that was kind of her original hypothesis with her data. So how do we do this? And so we designated about 17 different sites that we knew were streamside salamanders were located in 16 different sites. We talked to another professor at Middle Tennessee State University to try to arrange the sites on a variety of different kind of levels of biomass. And so we have sites that have very few animals as opposed to some of the most robust streams in the area. And we had a control stream that was located well outside the range, or located within the range that we knew that didn't have streamside salamanders, so we could show that it was detecting the animals where they were and where they weren't. And so we designated a 50-meter transect within each stream that we found that had good-looking habitat in terms of presence of streamside salamanders, either presence of slab rocks, nice riffles, and places for animals to lay eggs. And we designated kind of a starting point and an end point to the transect. And then prior to searching the transect for animals, Nicole would go and take water samples at the 0 point right here, at 25 meters, and also at 50 meters, which would be later filtered, extracted, and processed for environmental DNA. Now, upon getting the water samples, Nicole will do kind of extensive searches for both eggs, larvae, and adult salamanders. And so if you can imagine, this is a very kind of tedious task. Some of the streams will take between 4 or 5 hours to search 50 meters just because sometimes there's lots of eggs and lots of larvae. Well, she figured out pretty quickly that she couldn't count the eggs on site, so she would take pictures, take them back to the laboratory, and then count them one by one on the screen to kind of quantify the eggs. Larvae, she would do kind of these sort of removal searches where she would remove the larvae from the streams, from those sections. And on days where she remembered her data sheets, she would write it on the data sheet. But on days where you don't, you have to use your glove. But we do what we have to do, right? And so she made that happen. And then, of course, with the adults, we would capture adults, measure mass and snout vent length to get traditional measurements. Now, with the larvae, we weighed them in batches of 10 to acquire sort of a batch weight that we were able to kind of go back later and put on with biomass, and same thing with the eggs also. Now, what we did in each one of these 17 sites, we surveyed them 4 times throughout the season to kind of document early season, the midpoints in the season for 2 surveys, and then right at the end to see if we could document trends not only in biomass but also in eDNA. So after we fast forward a couple of different analysis techniques here, we started to see that there was a really clear relationship between biomass and also the amount of eDNA that's in the stream. Now, one thing we did was we ran several models where we included biomass in all of our models and then included a variety of other environmental parameters, things like pH, water temperature, and stream flow to correct for other things that might affect our ability to detect environmental DNA. And so along this x-axis, or the y-axis, this kind of equates to the overall kind of exponential copy number or amount of DNA found, whereas this represents the biomass. And so we found a really nice tight pattern along where the streams are, where the abundance is a bit lower, and so we're able to differentiate these lower abundant sites, but as we get kind of in the sites where the abundance is a lot higher, we get kind of much more disparate detections in terms of the overall abundance. So we're able to find a fairly loose kind of pattern between overall biomass and eDNA copy number, which is useful if we want to kind of do a rapid assessment of a stream to be able to quantify what's there. Well, the one thing we really found is that the variable that was most important for affecting our ability to detect was also stream flow. So we accounted for stream flow in these sites, and when I say stream flow, we're talking about this is what qualifies as a stream. And so we initially went into this project thinking we could use a traditional flow meter, but as you see, that's a bit of a problem. So then we tried to use the EPA's technique where you basically float a small orange down the stream. Well, as you can see, you can't float an orange there either. So we basically settled on cutting the top of a wine cork off and using that as our kind of standardized flow material. And so we quantified the flow over a period of several times to take an average flow to get a rough estimate of how that flow happens. And what we see is that as the stream flow increases, it decreases our ability to detect eDNA present in the sample. So in order to be able to derive an accurate estimate of, or somewhat accurate estimate of biomass based on the copy number, we need to account for the stream flow. That's the most important factor here. So then the question comes, all right, so there's a pattern there, so that's cool. Now what about, does it really mean in terms of life stages? Can we say anything about whether it's eggs, larvae, or adults? And so we tried to kind of look at a variety of things, and in a second I'll talk about that, but what we tried to do is actually look at the percent of relative eDNA that's actually present in the sample at a time. And after we kind of relativized streams by the greatest point in which that particular stream had the highest estimate of DNA throughout the season, we see that the optimal amount of time, if you want to derive kind of like a really good detection estimate and optimize your detection probability, it's somewhere right around the 25th of January till about the beginning of March. And so we're kind of right now in the window, given that season is still similar in terms of the weather conditions. Right now is the time that we need to be taking eDNA water samples. But then the next question that I was mentioning earlier is that, can we say anything about the larvae, the adults, or the eggs? And so we kind of went back and started looking at the days in which we detected these different estimates. We started looking to see which days we detected more larvae versus eggs versus adults at particular sites. And we fitted several models to these. We fit a linear model, we fit a null model, and a quadratic model, and we found that across the variety of groups, there's different times in which we think that the eDNA is coming from different sources. And so we think the adult DNA primarily occurs initially during the middle part of December, and that, through a linear model which fit this, about the maximum time, the last time we detected an adult was right around the 8th of March. If we see here, we see the actual eggs. Eggs are laid pretty highly early on, so most of the relative biomass occurs early on in this middle part from eggs. That decreases by about the 12th of April. And then larvae does maximize somewhere right around the 22nd of March there. And the reason I'm showing you this is if we take this figure and then combine it with the survey date, when we look at this kind of curve together, we can basically draw a line right down the middle which shows that when the larvae are hatching from eggs, this is the optimal period where a lot of this DNA is coming from to flow downstream. And so we think this was kind of a unique kind of study where we actually showed that, that we not only showed that a protocol really worked, but can also kind of loosely be correlated with biomass, but also we think it's due to the fact of the eggs releasing DNA when these larvae are hatching. So if you want to find a stream-side salamander based, if the conditions are the same as the year we did our work, Valentine's Day, baby, that's when it happens. And so the reason I'm bringing this up is kind of like a microhabitat scale type study is that we only did this survey on a 50-meter stretch in the stream, and we did it at 17 sites during one field season. So our inference is on a really small scale, and the inference is only based on that one season. We could go back another season, and these patterns may change somewhat. And so this is important to consider when actually trying to develop kind of conservation measures. And before I would tell like a monitor or kind of a state agency that the 14th is a guaranteed month, I would like to repeat the study across another couple seasons to make sure we're somewhere in that window of optimal sampling time. And so that was a project that actually turned out really, really interesting, which I was really nervous it wouldn't. I woke up many nights kind of scared for Nicole. So I was happy it worked out. And so we're going to shift gears and talk about another kind of important animal in my lab. We work a lot with the eastern hellbender. How many folks in the room here have seen an eastern hellbender before? A few hands, cool. All right, so for the folks that haven't seen an eastern hellbender, this is the largest amphibian in North America. These salamanders grow to, I would say, a maximum length of 24 inches. That would be just a monster animal, but most are going to be somewhere between 16 to 18 inches long that you would see for adults. These animals are extremely long-lived in the wild. They live somewhere up to 30 years in the wild, in captivity much longer. As you can imagine, looking at the animal, it looks very dorsoventrally flattened. They're adapted for benthic life. They live at the bottom of the stream, underneath big cover objects. They're fully aquatic. And so as larvae, hellbenders have gills, and so they acquire their oxygen as larvae through gills, but as they metamorphose into subadults, the gills go away, and they have these large fleshy folds on the side that allow them to absorb oxygen through cutaneous means. Interestingly, though, hellbenders have lungs. They don't use them very often, but they pretty much almost always rely on this cutaneous respiration in order to breathe. And as I mentioned, they're fully aquatic, so larvae and subadults and adults are all present in the same habitat in a very healthy stream. Now, what you'll tend to see is that in a lot of our streams, and especially Tennessee and other places where these animals have been declining, you'll tend to see very old adults that are large, but you don't see that recruitment of the larvae and the subadults. So that's kind of a thing that's been occurring range-wide. And so the one thing people always ask me is that, why do you care about hellbenders? Well, the one reason I care about hellbenders is because they're really cool. So, and they're the only representative of that family Cryptobranchidae, and so in North America. And so the name Cryptobranchidae implies hidden gills, and so the name crypto means hidden, crypto meaning hidden, branch meaning gill, and so it's a kind of a shout back to their name. This represents one of 3 species that occurs worldwide in this family, and so they're kind of a neat little thing. They're very old animals, and plus every time you see a picture of someone holding a hellbender, everybody seems to be smiling. So there must be something really good about them, especially kids. They really do like hellbenders too. And so I think about it as kind of like my role to maintain this kind of treasure of an animal here on this landscape, and they're obviously not doing well in many of the streams in which we have them. And so this is a figure that Kathy Jakowski actually has donated to several people that I know, so I'll stole that figure from you again, Kathy, so thank you for that. And so Kathy put together sort of a map about what's occurring range-wide across for hellbenders, and one thing you'll see in common is that you see a lot of states that have down arrows, which indicate known declines of animals, and also a few states where we kind of really don't know a whole lot about the abundance, and also some places where we think they're much more stable. This figure's a little older than, it's probably 3 to 4 years old, so there's data that probably needs to be updated on this, but I know Virginia has a little bit better kind of a handle of what's going on with their animals, and we're trying to get up to speed here in Tennessee. What you'll notice is places like Mississippi and Alabama, you see the X there, which indicates that their populations are generally extirpated. They will find a hellbender occasionally there, but that doesn't really say much about kind of a secure population. Now, if we look over here, we also see a disjunct population of hellbenders over here in Missouri, and you can see that estimates are there that populations are down by 80%. So range-wide, we can kind of see that hellbenders aren't doing well, or in some situations, we just don't have enough data to kind of conclude which places are stable and which aren't. And so a variety of the kind of research I'm going to be talking about now is going to talk about 3 different studies that I have going on in my lab or just wrapped up that have looked at various aspects of hellbender biology and conservation. Now, as I mentioned, if you have a really nice site, you have adult hellbenders, juvenile hellbenders, and larval hellbenders. This is a rare occurrence. Places like North Carolina tend to have a lot more of these than some other states. We have about 5 populations or 6 in Tennessee where we can legitimately find all 3 life stages. The one thing that's kind of a bummer about hellbenders is that it's really hard to take a good picture. As you can see in this one, it looks like they run the gamut of being a really nice hellbender picture or looking like fecal material. So it's very, very hard to actually have a good picture of a hellbender, but they're really, really amazing animals. And so what we wanted to do was we wanted to actually look at to see if we could quantify habitat use across all these life stages. There's been several publications that have quantified habitat use of adults and subadults, but in terms of larval habitat use, there's really just not a whole lot of stuff that's out there. And so one of my first master's students, he designated that as this is one of the study areas where he wanted to look into. And so the first thing first we had to do is that we had to identify 3 study sites in Tennessee that actually reliably had all 3 life stages. And these kind of span the north to south range across from the northern part of Virginia and Tennessee all the way down to where Georgia intersects with Tennessee down here. So we have 3 sites that are located in the Blue Ridge Escarpment here. And so what we did was we completed very extensive hellbender surveys at 100-meter reaches in all these streams. We did multiple reaches, and every time that we found a hellbender, we completed a detailed microhabitat assessment at the used site where we found the animal, and also at a paired random point where we completed the same type of habitat analysis to be able to compare what the animals were selecting by life stage, and also to see what's limiting in terms of a use availability approach. Now, what was kind of cool about this is occasionally we'd run across one of our friends while we're doing surveys. This actually occurred at Beaver Dam Creek, or one of the northern sites here, where we were actually doing some of our habitat assessments, and we happened to see a large animal that was out just cruising around in the daytime. And so periodically you get to see nice stuff like that. And so I'm going to kind of skip ahead with some of the major results here, but if we started looking at the different life stages, the one thing I will say is that we're only able to detect significant larval numbers from one stream. The other sites, we're able to get adults and both subadults. So for this analysis, we combined the larvae and the subadults together, and then also the adults separately. And what we found was pretty interesting. So both compared to both subadults and larvae and adults, we found that percent of large rocks greater than 256 millimeters and also the percentage of low embedded rocks that surrounded those rocks were very important for both life stages. Now what's interesting is that we found that the percentage of these kind of rocks that are basically the size of large cobble were important for larvae, but then also we included a metric where we looked at kind of larger rocks that were larger than 500 millimeters. And what we found out, that big distinction between the adults and the subadults and larvae was this presence of these kind of larger rocks along with low embedded rocks and also cobble rocks within that general surrounding area. And so that was kind of a really neat distinction that we're able to kind of show those large differences between these different life stages. But now the question still remains is, how does this really, what's really important for larvae? We really can't answer that question. And so we broke out the data from the one site where we were able to get reliable larval captures, which was one of the middle sites there in Tennessee there. And when we actually did a discriminant function analysis looking at the random sites versus larvae, juveniles, and adults, we were able to see clear separation from all three groups here, where we see the subadults down here, the adults right here, and also the larvae. And so we see this kind of differentiation among distance to bank and stream flow here, and also the rock size, going from larger rocks all the way down to more pebble and sand substrates. This X over here represents the kind of represents more of the sandy habitat, and this is where the larvae were found. So what we're finding is that the larvae tended to be more closely familiar of random sites that were available in the streams. And we explained this as this site where we find larvae has an extremely high density of larvae compared to other sites in Tennessee. And we think that the larval habitat is actually really abundant in the stream. It could be one of the reasons why we find such a high larval density compared to other places throughout Tennessee. Now, the thing that's interesting about the study is that we only did this at 3 sites in East Tennessee in one ecoregion. So we really can't expand our results across Tennessee until we start looking at other ecoregions and using these data to see if, in fact, high-quality habitat is available in other streams. So that's one thing to keep in mind. Now, a second thing we're kind of involved with right now is actually looking at sort of a population or sort of the population structure or the availability of high-quality streams throughout a variety of East Tennessee. And one thing we're starting to realize by looking at some of these river maps is that a lot of the previous locations where hellbenders used to be found historically are now found in TVA impoundments. And these impoundments were put in place with large dams for power generation in the early 1930s through 1950s generally. And the big thing that's upsetting about this is that a lot of our kind of well-known and well-established hellbender populations are fragmented by dam habitats. And so these animals can no longer migrate along the river corridors to be able to reestablish new sites or be able to have gene flow among these sites. And so one thing we're trying to look at now is trying to see what the efficacy of using translocation of moving animals from within watersheds to different streams that are isolated now, to see how animals do from being from a source site translocated to a new site to be able to maybe use as a means to bolster reproduction and potentially population structure. And you can see this from the diagram here in East Tennessee. Each one of these kind of red dots represents a TVA dam impoundment. So we see we have quite a lot of our waterways that are fragmented by these different dam structures. And so, as I mentioned, we're focusing our study in East Tennessee. This study is still currently ongoing. And so if you would notice the spatial scale, we're still operating at the microhabitat, but we're getting more into the home range of these animals. And so we use 2 sites as our source sites. These sites are known in East Tennessee as having robust populations. And we selected sites that have been declining in the past, but still have somewhat healthy hellbender populations. And we're trying to augment these habitats with these animals as they're translocated to these new sites. And so the 2 sites we're translocating animals to are Rough Creek, which is in the same watershed as one of our sites, and then Sittico Creek, which is in the larger Tennessee drainage. Right here, we're going from HIWASSEE River to Sittico Creek. And so the idea is that we're trying to use these animals to augment these populations that don't have as large of a population and see how the animals do if we need to translocate them in the future as a conservation strategy. So how do you radio track a hellbender? Well, you've got to find a way to put a transmitter around its neck, which is really hard, or the other way is to put a transmitter inside of it. And so luckily, we work really, really closely with a veterinarian. Her name is Dr. Rebecca Hardman from the University of Tennessee, and she agreed to do all of our transmitter implantation surgeries on these hellbenders. And so what we would do is we would capture animals at the site. We would use an MS-222 bath to briefly anesthetize the animals, and then we would sterilize the site with a little bit of chlorhexidine, rinse that off, and then Dr. Hardman would start her procedure, making a small incision near the lower body cavity. Insert a transmitter, which is a F1100 model transmitter by ATS that weigh about 4 grams, and then also implant a small pit tag inside the body cavity. The animals were sutured back together and then put in recovery tubs, which were placed in the river to allow clean stream water to wash over the animals until they had revived. Now, normally this type of procedure would be done ideally in something like a zoo, for example, where you have zoo-raised animals, where you can give them ample recovery time in the zoo. We figure by the time that we take the animals out of the stream, do surgery, move them to a vet facility, let them recover, and then move them back to the stream, it will almost be better and less stressful to release them on site. And so we decided after the animals were recovered, we released them right back to the sites. I will report that we had 0 issues or zero mortalities related to surgery after we completed the surgeries on site. So this was a very useful technique, and it seems to work pretty well. I was kind of concerned. I was worried the first time we would recapture the animals to see how they're doing if the incision sites would look weird. Their skin actually grew over top the sutures. So they actually were able to not only heal, but they actually grew skin way over top the sutures to kind of even prevent dehiscence from occurring. So we're able to track these animals, and luckily the way we did this study is that we did a post or pre-post design. So we surveyed these animals for a year at the sites in which we found them, and then the next year we translocated them to the new sites. We're able to have kind of a pre-post design to see how their habitat use is changing and also things like spatial ecology. And so this is Brad Neeson and Emily Nolan. These are the two master students that are working on this project. Brad is targeting more the habitat use and spatial ecology of the animals, and Emily is focusing more on the microbiome and skin health changes that occur with these animals pre- and post-translocation. And so both of these guys worked their butts off in the summertime. They drove between all these sites. Traditionally, they're trying to survey all the sites at least 2 weeks during the active season, so between May and August primarily. And then as we get toward more like October, November, they survey the sites once every 2 weeks. And then in the wintertime, the non-active season, they survey the sites once per month. And so they're able to keep up with the animals quite reliably, even before and after release. And so during our translocations, after we surveyed the animals for 1 year in the stream, and so it's kind of confusing, so we put transmitters in them, surveyed them for 1 year at the stream in which we caught them, and then translocated them to their new sites. The translocations occurred in May and June 2019, and in the end, we had 10 initial animals on Tumbling Creek, and we were able to translocate 4 males and 1 female. We've had some incidences with animal survival on our source site. We had one animal that was killed last summer by a fisherman who just stabbed the animal through the head. We found it on the stream bank later, which was kind of a really big bummer, as you can imagine. We had another female that died during one of the wintertime. We've actually had 2 females that have died during flood events in the wintertime, during what we think was last February, when we received about 15 inches of rain over a 2 or 3 day period. And so we think the floods kind of affected some of our animals in Tumbling Creek, but unfortunately we were only able to transfer 4 males and 1 female. Now I will say it would be ideal to have an equal sex ratio at the sites, but anyone who's ever tried to sex hellbenders, it's really, really hard. About the only time you can reliably do it is during the breeding season, and outside the breeding season it's very hard to tell what a male or female is. But luckily a researcher from University of Kentucky by the name of Paul Heim has developed a sex-based gene marker evaluation with PCR where you can actually tell if it's a male or female based on either a skin sample or even blood samples. And so we're able to use this to be able to sex our animals, but the unfortunate thing is you can't do it on site, so you're basically stuck with whatever you catch. And so turns out at Tumbling Creek, our first site, we were able to get 6 males and 4 females. And then actually at Tumbling Creek, we were able to translocate 6 males and 6 females, but the animals the initial number we started with was 16 animals, and we had a fairly equal number of male versus females. Some of the animals we couldn't translocate to Cedar Creek because we could never get them out of the rocks in which they were hiding. They were basically under big rock ledges, and then if you've ever done hellbender work before, unless it's a rock you can flip, the animals are really hard to get to. So we still have a few animals that are hanging out at HIWASSEE, so they're not going to get translocated because they picked a good rock to hide under. And so we try to translocate them in cohorts, so we try to do 2 to 3 animals at a time, just in case there was a large flood event that would occur that maybe would wash the animals out and cause mortality. We wanted to split the animals into groups of cohorts. And so we found some really interesting results. I'm just going to present the spatial ecology results so far. So at our one site from Tumbling Creek to Rough Creek, we found that the animals had very few post-translocation movements. When we translocated the animals and chose cover rocks for them, they generally stayed in a very small area. They didn't really move that much, which was fairly interesting. Now, if we look at some of the linear home ranges, they increased in late summer, which we think were males moving to find females to breed. But what was interesting about these, when we went back to do a survey about a month later, we found out that one of our animals, he was a large male, he put on about 75 grams of mass. And when we took the animal out to measure it, we stretched it along kind of the measuring board, and we squeezed on the stomach, a big mass of crayfish came out of its cloaca, which means he's just been snacking on crayfish nonstop. And so we picked a really good site for these animals in terms of food availability. And so the majority of our animals gained a good bit of weight. They didn't really move that much, which is cool. And if we look at kind of the other kind of linear home ranges before movement, the actual animals were moving more at the source site versus the translocation site, which is fairly interesting. But now let's say we look at our second site here. This is the HIWASSEE River to Cedar Creek. We found out we had an average linear home range of 688 meters after translocation. So we're talking about animals that are making very large movements. You can see we had one animal right here. This was one of our main release sites. The animal moved all the way down here to this river bend, all the way back up again, all the way up to here, and made a very large series of homing movements. This happened across several of our animals, and we also found at this site the animals in general were losing weight upon the resurveys. So it doesn't appear that the animals are doing quite well at the site. And if we look at kind of the graphical representation of this, we can see that those linear home ranges and kind of the 25% core home ranges are increasing considerably here. Now, what's interesting about it is we lost quite a few animals to predation at this site. There's apparently a very large established population of river otters that we were unaware of. And if you look at right here, we have several transmitters that are indicated in kind of this red dots right here. The otters kind of made short work of about 6 of our animals at this site, so this was kind of a big bummer. This site, the animals were surviving very well until about August, and then we lost about 6 animals in a period of a week. And we think this is attributable to the males making large movements for potentially finding breeding rocks and being vulnerable to attack by river otters. This is a site that actually has a lot of trout stocking, so we wonder if, in fact, there's some subsidization with the stock trout for maintaining really large otter populations. And so after this fact, we're pretty upset. Everybody's pretty sad, which is kind of a bummer. But we put out game cameras to try to see if we could target the otters that were doing it. And in fact, we saw pretty convincing photographs of otters out at nighttime, which probably explains why we don't have hellbenders there right now. Well, I will say we have 4 animals that still survive there, but we lost a significant chunk during this kind of big predation event. And so what did we learn from the study so far? Well, Brad's still looking at a lot of the spatial ecology data to kind of put together official data for that. And he's just now delving into the microhabitat results to get some data on that. But overall, we can kind of conclude that at one of the sites, we saw larger moose post-translocation. And we think this is due to kind of the general stream differences between our source sites and translocation sites. Tumbling Creek and Rough Creek are very similar in terms of the overall gradient of the stream and also the size of the stream. And so we think that that's kind of a big thing where the animals are being placed into a stream that seems familiar to them. As opposed to Sittico Creek, which has really nice cover rocks and really nice habitat, it's a faster flowing stream. It's a much smaller stream, and we think it was more of a shock to the animals upon release to be able to kind of do homing behaviors to find familiar habitat. Secondly, from anecdotal assessments, we can just look at the stream, and by flipping cover rocks, we know that the crayfish density is significantly higher in Rough Creek compared to Sittico Creek. So we think a lot of the animals just not moving is the fact that they have available prey sources underneath the rocks. In fact, one of the times when we were recapturing during health surveys, one of the rocks we flipped up, there was a large male hellbender that we translocated, and there were 4 crayfish under the rock with him. So he's just like literally just sitting there waiting on something to walk by. And we think the similarity of the habitat along with the crayfish density is probably a reason why these animals are doing much better at Rough Creek compared to Sittico Creek. But there's going to be more data that's going to come out of that hopefully soon. And so to kind of go out a little bit in spatial scale in terms of our hellbender work, this is the kind of the big research that Geronimo did, my first graduate student, when he was a master's student with me. And one of the big questions we still have to ask ourselves is in Tennessee, if we kind of go to the larger scale, like, do we know where we even have hellbenders? We have an idea where we have some healthy populations in East Tennessee, and we know we have a few things kind of scattered throughout Tennessee. But if you look at Tennessee in general, there's 13,000 miles of stream. So there's a lot of potential habitat where a hellbender could even exist. Can we develop a way where we can actually try to model this habitat and then do on-site assessments to assess our model to see how accurate it is? And to give you an idea where hellbenders occur, basically anything to the right of this red line is where hellbenders occur in Tennessee, or potentially. So we're talking about a lot of stream miles here. And so one of Geronimo's initial goals was to actually develop a distribution model for hellbenders in Tennessee. And he used a variety of things like distribution data, environmental information, and also things like presence-absence data and a variety of different environmental data to come up with an estimate of that overall range projected across the state of Tennessee. Now, as you'll notice, this study is occurring strictly at the landscape scale. So we're trying to develop an overall kind of habitat suitability model for the state of Tennessee based on using the MaxEnt algorithm and then provide an on-site assessment of the streams. Now, if you've ever done hellbender work before, you know it's extremely exhausting and it's really, really hard. A lot of times you'll survey a stream for 5 hours and then not find an animal. But the problem is with this is that you could still easily have an animal there. So being able to come up with a quick way to assess presence-absence is difficult. So therefore, we employed some more environmental DNA to use as a potential tool to kind of provide a quick assessment of these stream environments. And so basically this model, this doesn't look very pretty, but this is kind of the output from MaxEnt here, where Geronimo coded the streams on a scale between 0 and 4 using natural breaks in the data. The zeros aren't shown here because those are fully unsuitable streams, but on kind of a gradient of low, medium, and high, and very high suitability. And then we developed a series of different kind of stream stratifications to be able to survey across this gradient to see how well our model worked. And in the background, we're always hoping we can find our little devil dog, which is one of the colloquial names for hellbenders here. And so that's the idea. We're trying to find our hellbenders. And so initially, this was some of the initial work for environmental DNA that occurred in Tennessee. This was done by Dr. Michael Freak and Dr. Stephen Spear in the years of 2011 through 2013. They surveyed about, I think it's somewhere around 80 to 90 sites in the state. And so we used some of the preliminary data to kind of guide our selection of sites. And then we added on top of that an additional 200 sites on top of that to actually acquire environmental DNA samples. Now what you'll notice is some of the sites are labeled in red. These indicate multi-sample sites where we would go back seasonally and take multiple samples at the site so we could acquire an estimate of detection and our final estimate of overall occupancy. And so as you can see, we had 200 sites total and then 50 sites across that gradient of that 1 to 4 sample. So we excluded all the sites that were listed as unsuitable, but across the sites from 1, 2, 3, and 4, we stratified 50 samples across that. And then, of course, the red, like I said, represents the seasonal sites. We tried our best to sample about three times per year at these sites. And at every third site, we also acquired a negative sample, so we filtered deionized water through our filters to be able to kind of assess our ability, how clean we were with our assessments, and to kind of control for the effects of contamination in our samples. So what did we find? We found a total of 65 sites where we had positives for the animals and a total of 226 negatives. So we're definitely on the side of not finding a lot of animals. If we did naive occupancy, that's about 22%. But our detection probability, which is 55%, which is actually a little on the lower side compared to other studies. But then after we corrected for this, our corrected occupancy is somewhere around 45%. Now, this is quite interesting because when we started looking at our sites across that disturbance gradient, we found that a majority of our detections were coming from the sites that were modeled as the highest quality habitat. So we feel our distribution model is somewhat useful in the fact of being able to predict our sites that are of the highest quality down that gradient of site suitability. Well, one of the things we modeled when we looked at our eDNA detection, we looked at Julian date. And so Julian date in our study didn't really appear to play a big role. And the reason this is important is that hellbenders have a very clear breeding season when they breed in August and September. If there's males present, they're usually kind of either fighting with each other, they're releasing sperm into the stream, they fertilize eggs much like fish do. So you would expect that eDNA would be very high this time of year. And a study by Stephen Spear, he found that there was a really clear pattern of this kind of seasonal effect on the ability to detect a higher amount of eDNA at this time of year. We think this has to do with the fact that we ranged our sites across a variety of suitabilities. So we had really low quality sites mixed in with the higher quality sites. So we think we're missing, there probably are some of the animals at some of the lower quality sites. We just don't have the accuracy with the estimate to be able to pick up those animals. And one of the biggest issues of environmental DNA, the dilution of your water sample plays a big role. So these larger stream environments that may have a lot of water and very few animals, you're under extremely high risk of missing animals there when they're present. So we acknowledge this and see the fact that our detection probability isn't phenomenal for sure. Other studies that have come out that have been at sort of better stream environments with much higher hellbender populations are as high as about 90%. So we're definitely on a much lower side in terms of the overall detection probability. So we started looking at occupancy. When we corrected for detection, the most important parameter in these models were ecoregion. And so when we start looking at ecoregion, we could see that we can gain some kind of an idea of the stronghold populations. And we see kind of two general patterns here where we see the Blue Ridge escarpment, the Blue Ridge ecoregion right here. This is kind of the border between Tennessee and North Carolina. We see that the majority of our detections occurred along this and also in West Middle Tennessee along the Big Swan, Little and Big Buffalo rivers and a few sites that are scattered that feed directly into Tennessee. Now, one thing you'll notice is we get periodic detections across Middle Tennessee, which is interesting. But one thing we also see is we see a lot of negatives across this area also. And so when we looked at the overall occupancy, we can see that the Blue Ridge is quite high, almost in terms of the realms of about 0.75. The lower side is the interior plateau ecoregion, the Ridge and Valley, and also southwestern Appalachians are definitely considerably lower than both the Blue Ridge. And so Geronimo and I kind of talked about some reasons of why this is. And so Geronimo went an extra step further, and he created a watershed use map across Tennessee, where he looked at both the prevalence of agricultural and urbanization activities and created sort of an overall score for the Huck 12 watersheds. And if we look at these ecoregions, we can see some really kind of clear patterns. The interior plateau had the lowest occupancy. And what we notice about the interior plateau is that we have quite a considerable amount of red and orange, which indicates lower quality watersheds. And this is primarily due to agriculture in this ecoregion. We also have quite a lot of agriculture in the Ridge and Valley ecoregion right here also. One of the ecoregions we actually didn't do any surveys up here. It was in the northern Appalachians here, and also the central Appalachians, which is the Cumberland Plateau. A lot of the habitat just really isn't suitable. There's not a lot of larger streams there that would hold hellbenders. But we see we have high-quality habitat. I just don't think we have a high quality of suitable habitat in terms of stream order. Now over here, we also notice in our places where we have kind of clear populations still, we see fairly intact watersheds. Definitely a little bit more orange compared to something like the Blue Ridge, but we can see kind of a clear pattern of land use, which is impacting where we're finding our hellbenders. So this kind of goes in with what a lot of researchers that are saying that in fact that land use and also erosion and subsequent sedimentation appear to be a big problem with hellbender populations. Now, the one thing that's kind of interesting with the study is that we still don't have a link between all three of these studies I've talked about. And the ideal study design would be to design kind of an eco-region-wide thing where we assess microhabitat and also kind of larger landscape patterns to be able to kind of fill in some of the micro-scale stuff along with these larger landscape scale pictures here. And so that's kind of where our hellbender research is at in Tennessee right now. We're starting to get kind of a good idea where we're finding animals, but the idea now is to try to understand where we have suitable habitat and potentially restore sites that would be suitable for translocation of animals from zoo-raised animals or potentially from robust populations. So that kind of provides that connection of how all three of those are sort of linked together. Now, as we kind of shift gears one more time, I'm going to go up the spatial scale just a little bit more. In this example, I'm going to talk a little bit about some of the research that I actually worked with Dr. Barrett here whenever I was hired here at Clemson as a postdoc. And the project that I was brought on to look at was to not only help designate these areas known as priority amphibian and reptile conservation areas, but to understand what the impacts of things like climate change and also projected habitat change or future habitat change may have on these areas. Now, if you're not familiar with what parkas are, they're basically these areas that target areas of very high endemicity of amphibians and reptiles or very high exceptional species richness of populations. And so this would be similar to something like the important bird areas. If you guys have been out at a park and seen a sign for an important bird area, the parkas are supposed to mimic this type of a process to kind of target these areas of, or highlight these areas of exceptionally high amphibian and reptile biodiversity. Now, the study scale of where we were working, the extent here, if we look at, this is Virginia here all the way up through Maine. So we're working basically in the entire eastern seaboard over here in the northeastern U.S. I've recently completed this process helped complete this process for West Virginia, so we can now fill in this gap here to be able to produce a map of parkas and also potentially look at things like vulnerability across this landscape. Now, the idea is that we want to kind of target these areas so we can get areas we can know areas for our biggest conservation bang for our buck. We can look at the potential protection of these habitats and then potentially leverage the fact that these parkas are areas of importance to maybe garner future protection from things like land use. But the problem is that, as I mentioned, we don't really have any kind of idea of knowing how these areas are going to change in the future. So the primary role that I played in this project was to develop a vulnerability index and use this across the landscape to target which habitats are most vulnerable throughout this area of the northeastern U.S., and also to see which parkas are potentially most vulnerable to change. Now, when we developed this vulnerability approach, I thought about it from trying to do sort of like something similar to the vital signs program by the National Park Service. We wanted to use a multi-metric approach, which is very similar to something like a doctor's visit, where if you go into the doctor, the doctor listens to your blood pressure, listens to your breathing, gets measurements on your weight to try to get a multi-metric assessment of overall health. And so we wanted to think about developing these vulnerability indices from a perspective of multiple variables, so things like potential occupancy of rare species, maybe presence of invasive species, for example, percent urbanization, and overall habitat condition. And this would be potential variables that we could assess at our site given the data that we had to be able to do that. And so to kind of further fill this vulnerability assessment out, we did it from a three-angled approach of looking at things like exposure, sensitivity, and adaptive capacity. And exposure is basically the explanation of the extent of an environmental stressor that a species or landscape experiences, whether we're talking about relative climate change, sea level rise, or a variety of other stressors. Sensitivity, on the other hand, refers to the degree to which a species or area is impacted by a stressor. So you may have a particular landscape that may be fairly sensitive to change, or particular species that are there that are also sensitive to things like climate change. And then lastly, we have something what's known as adaptive capacity, which refers to the capacity of a species or area to cope with a general stressor. So one example might be if you have an environment that has really high elevational relief, there might be a variety of climatic niches that species can move to in response to climate change. That would be an area with high adaptive capacity. But the issue is that there's a lot of things that can be used to fill in these estimates here. And so that's the big question, is what types of data do we have and what can we use to be able to derive estimates of exposure, sensitivity, and adaptive capacity, and then combine those to an overall estimate of vulnerability of that ecosystem. And so after Kyle and I met several times, we discussed a variety of different types of data measurements. We kind of put together what I like to call a GIS sandwich right here. This represents the kind of spread of different data layers that we potentially thought we could use to assess a different aspect of ecosystem vulnerability. And so we used a variety of data sets, including things like land use land cover data, natural landscapes, protected areas data layers. We ran a series of distribution models for species, and a variety of other metrics that would estimate different aspects of both exposure, sensitivity, and adaptive capacity. Now, if you think about it, from the top layer here, you see these kind of draft polygons. The idea would be that we would extract data from each one of these different layers to derive information about overall exposure and things like other submetrics and overall vulnerability for each parka and each pixel across the landscape. And so one of the kind of important kind of wrinkles that we put into this was sort of a really kind of nuanced approach to look at a variety of amphibian and reptile species and their potential response to climate change. And so we used a kind of a large, really robust approach to look at a variety of species. And so, for example, in this example, I'm going to talk about green salamanders. And what we did for each one of our priority species, we used a climate envelope modeling change approach using Maxent modeling from current to 2050. When I conducted this analysis, this was right around 2014, so it would be from 2014 and looking at changes all the way to 2050. And we used two global climate models, including the CCSM4 and the Hadley GM2 model. And the issue with this is that there's a variety of climatic models that make a variety of assumptions. And so from some of the reading that we've done on past manuscripts, it appeared that these two climatic models predict pretty well in the northern latitudes, which is where our studies were located. So we thought these would be kind of really nice general circulation models to start with. Then also with this, we used two RCP scenarios, which will look at greenhouse gas scenarios, with the 4.5 and the 8.5 as being kind of two kind of moderate and also extreme greenhouse gas estimates to be able to bracket our estimates of the organismal responses to climate change. We used a set of uncorrelated bioclimatic variables to do this. Then we used three thresholded approaches to get kind of more kind of general to conservative estimates of what the animals' change over time might be. And then we ensembled the RCP and 8.5 levels together to come up with sort of current levels based on the 4.5 RCP and the 8.5 to look at the reduction of the climatic niche of the animal over time. Now, when I say that the animal has a reduction in climatic niche, this doesn't necessarily mean the animal is going to die because of climate change. We're just thinking about the broad scale. At the kilometer scale, is that climatic niche going to be the same based on these two scenarios in the future compared to what is currently available? And to give you an example of what this looks like, this represents the current climatic distribution of for the green salamander. I've buffered the range by about 50 kilometers on either side. So we can see that we're, based on the current data and where we have these animals now, what the kind of current climatic niche or kind of the accepted climatic niche is for the animal. Now, if we start looking at the 4.5 scenario, we start to see a little bit of reduction in these kind of more kind of climatically suitable areas. And also in the 8.5, we see a larger reduction. And again, I want to say this doesn't necessarily mean we think the animals are going to die like immediately from climate change, but there's going to be some changes in what is considered to be their current climatic or optimal niche. And so we use these as an additional estimate across the variety of amphibian and reptile species to derive an overall change in the overall climatic niche of these conservation priority reptiles and amphibians. And we repeated this process over, I think it was 61 priority species. So we did this for quite a lot of the animals in the northeastern U.S. And so to kind of put this all together, so you obviously have estimates of things like exposure. This is what's represented in red. Yellow represents sensitivity, and green represents adaptive capacity. Now in this example right here, I'm going to use landscape integrity as our stressor. I'm going to use range losses priority species as the sensitivity metric, and percent protected areas as our adaptive capacity metric. Now what we did was we scaled each one of our variables on a scale between 0 and 100 in GIS, and then we derived an overall average across each one of these kind of primary metrics across each one of these submetrics. So for example, we used landscape integrity, the climate change estimate, precipitation change, and sea level rise where it was relevant to derive an overall exposure estimate as a synergistic estimate of exposure. We did the same for the sensitivity metric and also the same for the adaptive capacity metric. And as I mentioned, averages here. And so then to calculate resiliency, we subtracted sensitivity from adaptive capacity and use this number for resiliency to come together with the average from the exposure metric and to come up with an overall estimate of vulnerability by subtracting resiliency from exposure. And we did this for every raster cell across the landscape, which was at a data grain of about 300 meters across this northeastern landscape. And so the cool thing is every ecologist has to put a math equation up there to make it look complicated. So this is what that looks like. So that makes me feel better. And so we kind of look at each one of these metrics separately. So if we looked at the exposure metrics, we can derive a variety of information from these kind of metrics across the landscape here. We have things like sea level rise, which is hard to see, but the Chesapeake Bay area is predicted to have the largest impacts from sea level rise. We see kind of the northern portion, places like New York, are going to have the largest change in terms of temperature. Precipitation was a weird one because I expected some places to get drier and some places to get wetter. But what I found was that overall is that the Northeast is predicted to be just wetter in general. And so we found kind of the higher elevation areas are going to receive higher rainfall than what's normally found now. And then lastly, things like natural landscapes. This is a prediction into the future by 2030. We can see that the Mid-Atlantic corridor and places like I-90 corridor are projected to have the largest impacts from things like anthropogenic disturbances. So when we put this all together, we get a basic map that looks like this, where we see that the largest exposure is going to be along this Mid-Atlantic corridor here all the way up into places like southern Vermont, for example. Now, if we go look at the sensitivity metric, we have our amphibian and reptile climatic change layers that we developed from our 61 species. And then we also kind of put in a measurement for what were known as our Tier 1 conservation priority species. These would be species that are either state endangered or federally threatened and endangered. And so we developed this as kind of the species that would be potentially most sensitive to landscape change over time. And we put these together to come up with the overall sensitivity metric with western Pennsylvania and the central portion of the Mid-Atlantic corridor up through kind of southern New York as having the highest estimates for sensitivity. Now, if we looked at adaptive capacity, we looked at things like landscape permeability, elevation standard deviation, protected landscapes, so places that are either national forest, national parks, or state preservation lands. Looked at things like the habitat diversity metric, distance from ecoregion boundaries, and also things like hillshade to be able to derive an estimate of overall adaptive capacity. And what we see are that the places where we see kind of the largest elevational changes and the places like where we have a lot of protected habitat, places like north central Pennsylvania, the kind of highlands here, the White Mountains, the Adirondacks, and also parts of Maine, tend to be the places where we see the largest adaptive capacity. Again, we see kind of low adaptive capacity in places like the Mid-Atlantic corridor and also areas surrounding upstate New York here. So when we finally put this all together for our vulnerability metric, we see kind of some interesting trends pop out. We see that the highest vulnerable landscapes are kind of located in this north-central area along the Mid-Atlantic corridor, western Pennsylvania, and along these areas, where places that have a lot of elevational protection, elevational variation, and habitat protection tend to have the lowest vulnerability. Now, the idea of this was to relate back to the parkas. I thought it would be best to have a continuous landscape, so we would basically have to take our polygons and extract data to be able to derive those data for each one of our parkas. And so if we kind of look at our parkas projected across the landscape, our vulnerability, when we divided it back down, it ranged between 2.24 to 5.86. It's supposed to be on a scale between 0 and 100. I forgot to change that on here, but what we found out is that our most vulnerable parka was somewhere around in the upper 70s, so we didn't actually have a parka that had 100% vulnerability. And our lowest one was somewhere right around the low 20s or low teens. And our most vulnerable parka was found up here around kind of this area of New York, and our least vulnerable parka is this little guy right there on the kind of western terminus of the highlands of Virginia. And as I mentioned, the areas like the western Allegheny Plateau, North Atlantic Corridor, and the Great Lakes regions are the areas that have the greatest vulnerability. Now, what's cool about this approach is we can actually essentially give a report card for each parka if we need to do that. And so basically we can kind of score these metrics on whether they have low to high vulnerability, exposure, adaptive capacity, or sensitivity. And so, for example, if we take our most vulnerable polygon, we found out that the projected natural landscapes indicating that increasing anthropogenic threats are probably the biggest threat to this landscape. Also, things like in terms of adaptive capacity, this is a very flat landscape, doesn't have a whole lot of elevation variation, very few protected areas, and is very close to an ecoregion boundary. So we can use these different estimates to come up with management strategies for each parka and potential ways in which we can increase the conservation status of each one of these landscapes. Now, I will say I don't really want to do this for each 650 parkas, or however many they are, but if a manager has a particular question of each specific parka, I can derive these data to be able to produce that report card for him or her. And so overall, we came up with a metric that does provide stakeholders with a conservation decision tool for vulnerability. It does incorporate multiple monitoring objectives as necessary. So initially, these were the kind of objectives that we thought were most important for predicting amphibian and reptile presence and also kind of persistence across the landscape. It does provide multiple levels of biological information that can be changed if need be, if there are better variables or better data sets that can provide better information. This can be swapped out for another data set and used quickly. It does not imply biological importance based solely on vulnerability. And so what I mean by this, let's say, for example, we have a highly vulnerable landscape, but it's because we have a really high presence of endemic or Tier 1 conservation species. And so we need to evaluate the reason why it's very vulnerable. It could be the fact that it has a lot of species that are extremely important to protect, as opposed to something like climate change affecting it. And so we need to know really what's causing this kind of high vulnerability score. It does not fully appreciate the climate buffering effects of microhabitats. And so again, When we did some of the initial kind of vulnerability assessments, we did this at the 30-meter scale. We eventually had to sum up to the 300-meter. But it doesn't really appreciate, even at this scale, the climate buffering aspects of microhabitat. And what I mean by this is that if we have, for example, a small salamander, like a green salamander, or something like a Shenandoah salamander, they're using a 6-meter area for their home range in a given year. We're obviously way beyond that with our ability to estimate that climatic niche. And so when I talk about losing the climatic niche, I'm not talking about losing the species. I'm talking about the overall change in the available climatic parameters from what's witnessed now as opposed to what's happening in the future. So we primarily want to use this estimate of climatic niche as an estimate of vulnerability and not so much as persistence, if that makes sense. It does not predict species loss, just loss of climatic niche, like I was just saying. And the way we initially designed this metric, this does not weigh it considers all the metrics similar. So they're all on the same scale and aren't weighted differently based on the type of metric. So if a given manager, if we wanted to, or if a manager, or for example, like something like the Northeastern Atlantic or the North Atlantic Landscape Conservation Cooperative wanted us to weight something like landscape change much higher, we would have to rearrange the metric to account for those differences amongst those metrics. So right now we're considering all metrics equally upon the vulnerability of amphibians and reptiles. And so the idea for this thing is to kind of wrap this project up. We're kind of in the finishing stages, and I feel like we've been there for maybe a year and a half. So the idea is to get this project finally wrapped up and completed here, but we're really close now. And so I'm going to kind of end the seminar with a few of my final thoughts here, kind of a nice conclusion slide to wrap it all up. Understanding of spatial scale is essential for correct inference. And what I mean by this is that it's really important during the initial phases of project planning is to think about how am I going to use my data, what types of questions can I answer with this data, and is my data really going to achieve the objectives I have to start off with? And this is something I usually sit down and talk about with my graduate students before we even start a project. Secondly, as spatial scale increases, ecological mechanisms generalize. And so it's important to kind of keep this in mind as we start going more of a landscape approach. We understand fewer and fewer kind of overall mechanisms. This kind of translates to more kind of larger scale type processes, things that operate more at the ecoregion or even kind of the biome scale, for example. Hierarchy is important for understanding mechanisms and limitations. And what I mean by this is that if you're doing a landscape scale approach, it may be useful to kind of have a few sites in which you do intensive surveys or really kind of do intensive data in order to understand more about a mechanism as opposed to that overall landscape approach. So being able to nest sites or kind of more intensive surveys within larger landscape sites is an effective way to gather more data. Development of conservation strategies that leverage all spatial scales is important, but as you can imagine, it's extremely challenging. There's reasons why we don't do it. It's because it's extremely expensive. It requires a lot of work and a lot of time and a significant amount of funding. But if you're able to pull it off and have the help to do it, that's obviously the way you want to do it, to incorporate all spatial scales into your study. And lastly, this is what I say to everybody: get out in the field, you might learn something. And this is something from like my graduate research and even across the stuff that I do now. Still my favorite part is getting out in the field and observing ecological properties and getting out with my students to work with the animals that we all love and want to conserve. And so with that, I want to thank the TSU Wildlife Ecology Lab, which is a lot of great different students. I've had a variety of master students that have worked in my lab already, a PhD student who's going to be finishing up this summer, and various undergrad students that have helped out with a variety of projects. I want to thank a lot of the collaborating university and also nonprofit partners that have helped out a lot with this research. As you can see, there's a lot of people involved here, and I obviously can't do it all by myself. And then lastly, I want to thank all the funding sources that have provided funding for us to do our project and research. So thanks for your attention, and I'll take any questions.
Questions for Bill?
Yes?
Back like 800 years ago, you showed the map of like the streams in Tennessee,
Mm-hmm.
and then you threw out that red line
Yep.
of like hellbenders exist east of this. How do you know that? Because you also have that Missouri population, so...
Yep. So I'm just talking about specifically in Tennessee. We're talking about just in Tennessee. They're only associated with Tennessee River drainages. And so at that point, that's where the Tennessee River goes down in East Tennessee and then comes back up in the west part of Tennessee. So the populations are just found within that general kind of flowage of the Cumberland and Tennessee rivers, which eventually go to the Ohio. So that's kind of like, and when you see, the reason I put that up there is because when you see a Peterson Field Guide, it shows you the range of a hellbender, and it's like they're in this blob. And obviously hellbenders are not everywhere because they're really hard to find. And so I like to think about it from that perspective. It's kind of a misleading map. So, yeah. Yeah.
Curious, you were talking about the stream size elevators, and you found less DNA where you had high stream flow. And you may have said this and I didn't catch it, but is that because the DNA is harder to detect because of the increase in discharge, or is there something about that increase in discharge that you have fewer salamanders in the stream?
I think it's related to the detection of, I think there's DNA that's moving quicker down the column. And the problem with eDNA is that I think it's not a continuous thing. Like, I think there'll be kind of clumps of it that kind of come and go. And with some of our stuff, I didn't talk about this, but sometimes at some of our sites we take 5 samples and we would detect detections with 2 of the samples and not the 3 others. So I think sometimes it gets a little bit more difficult to get that clump of DNA in that water sample you're taking. And I think as stream flow increases, it probably dilutes it out a little bit more, or potentially makes it harder to pick that up. So I think it's kind of one of those things where there's, at the sites that are super abundant, it may not be as much of an effect, but I think the lower abundant sites, it probably makes it much more difficult to pick that up. But that's just kind of what we're hypothesizing. But that's the issue with eDNA, is that it still requires, I think, a lot of laboratory experiments with kind of setting up little raceways with kind of putting in known amounts of DNA and increasing stream flow are really what we have to really understand those kind of mechanisms. Yep. Yes?
Do you think that if that is the case, and either stream flow or amount of rainfall or whatever it is going to affect your ability to detect them more, do you think it would be worthwhile to, instead of modeling your detection by date, model it by like amount of rainfall that you've gotten in a period, or like overall stream flow or something like that?
Yeah, so
I'm not an aquatic ecologist. So.
No, that's great. No, that's a great question, and I think that's definitely something that would probably be a valuable asset to kind of look more just beyond like day of year, because like we're looking for just more like when we kind of take it even back to not even trying to do abundance, but just like did we find the animal on that day or not, or detect it. We even see a seasonal trend with just straight binary occupancy. When we try to map that, it's like clearly like that kind of date between like, mid-January through like the beginning of March is still like the optimal time. But I am sure things like flow after rainfall really, if you think about it, like there'll be like a big flush of water that moves through there that may actually move DNA that's even stuck to substrates and things like that. So there's a lot of things that are problematic with eDNA, and that's definitely one of them for sure, no doubt.
Past the hour, so we'll stop it there. But again, you guys are all welcome to come to the house this evening, and let's thank Bill one more time.
If I go a little long, what's it? Rather long.
They do it a lot, like some of them
Instead of like
the situation is not as dire with the Eastern Public Service, so like Ohio, kind of population.